Hi everyone, welcome back to Rocks for Jocks. My name is Alex and I'm your host for the show. I have a really fun episode for you all today, as this was the first episode where I brought my show on the road. It's the first time where I physically recorded somewhere besides my own apartment. So I brought my microphones over to Harvard's campus and had an awesome chat with a PhD candidate over there, Amanda Calhoun, who you're going to hear from shortly. She works in the Department of Earth and Planetary Sciences. So really excited for you all to hear about that and see the awesome research that she and her professors are doing. I'm recording this towards the end of April, where Harvard University is currently in a little bit of a limbo state in terms of their certainty that some of their research is going to be able to continue. I personally think that it's more important than ever to publicize their research so that people can understand more about what they're doing and why it's important. This interview, of course, only covers a few projects being conducted by a single PhD student, and Harvard does a ton more, including lots of medical research and teaching, which affects a lot of us in the Boston area directly. Hopefully this episode showcases what I think is at least one example of some great research that they're doing and why it is worth continuing to fund it. If you're listening on Spotify or Apple Podcasts, please rate and review the episode, as it helps this channel climb the ranks and reach more listeners. And if you enjoy the episode, please send it to a friend or maybe send it to your family group chat to spice things up. It'll give you something really fun to talk about next time you see them. Thank you all for listening. Now enjoy the show. Amanda, thanks so much for joining me today. One of the things I like to do to start is kind of give everyone a chance to talk about themselves for 30 seconds. Maybe just tell me a little bit about who you are, what your role is here, and how you got here.
Well, yeah, so I'm a second year PhD candidate now. Yay. In Earth and Planetary Sciences, specifically working in geochemistry is my focus. In my undergrad, I double majored in chemistry and earth science, and am now applying isotope proxies to the past to try and understand Earth history, and then sort of getting into more of origins of life stuff, and paleoproxies. So yeah.
Perfect. Yeah, I saw on the Harvard website at least that there was a little bio about you. The quote was pretty long with a lot of big words, so I'm not going to read it out completely, but my understanding is that it's you're working on basically chemistry to understand different forms of life. Do you want to maybe correct me on that or expand on it at all?
Sure, yeah, I guess, yeah, because I was a chemistry undergrad, I have that sort of background, but the research that I got into in undergrad and that I'm working on now is super interdisciplinary, I would say. I have two projects that are not super related to one another because I am co-advised in grad school. And one of them basically tries to use carbon isotopes as tracers of life in the past. So if you went to some of the oldest rocks on Earth and you applied carbon isotopes to them, could you say whether or not life was present and produce that signature? And then I have a completely different project, which tries to figure out the mechanism of this chemical process called pyrite oxidation. So it's weathering of the mineral pyrite, which people know as fool's gold a lot of the time. And yeah, this is a really important process for understanding Earth's oxygen history. So Earth was not always oxygenated. And then also for understanding modern acid mine drainage systems, which produce a lot of acidity and oftentimes need remediation for those ecosystems.
Cool. Yeah, the pyrite in particular, I think, is really fun to mess around with. When I was a TA in grad school, one of our favorite weeks was basically passing out pyrite and a bunch of other samples to students and have them scratch it with, I don't know, nails or other rocks or razors and try and see what they could scratch out of different stuff. And pyrite was one of the class favorites, I think.
Oh, absolutely.
Just for having that fool's gold, the shiny qualities and all of that
exactly.
stuff. Yeah, so tell me a little bit more about that. How does that process work? How do you start to figure out what chemicals are in rocks or have been in rocks in the past?
Yeah, well, there are so many different subdisciplines in this field in geochemistry, I would say. And so my background is really in looking at lipids or fat molecules that are in natural systems. And I've just transitioned now into using isotopes as tracers, which isn't really the mineral composition of the rock, but it's more on the atomic level. Do you have an oxygen 16 or an oxygen 18, which is getting really into the weeds, but it's basically how heavy are the oxygens that are in your rock, I would say.
Yeah.
Or your resultant product. So nowadays, even though I'm like a geologist, I don't really look at rocks very much. I look more at like natural waters or sediments in hot springs or something like that.
Oh, really? Why do you go to the waters themselves?
Yeah, the waters contain like the dissolved products of the process that I'm looking at. So if you have pyrite that is then weathered, it produces this compound called sulfate, which is dissolved in waters. So if you sample the waters, you can kind of get at that product material that you're interested in. For my other project, I study the microbes that live in hot springs. So it's like you scoop up a bunch of sediment or a bunch of water from the spring, and you can study the community that you're interested in.
How have those projects gone for you so far? Have you actually gone out and taken the samples and started that process?
Yeah, so for the hot spring project, I kind of got into that type of work in undergrad. And so I did go to Yellowstone in 2022, and I'm still using samples that I collected from that project. We have really cool collaborators there from Montana State who obviously have easier access to the park. A lot of times they get samples. But that was a really cool field expedition. And I did use that material for some of my undergrad research, which was trying to understand how microbes adapt to extreme conditions, because those hot springs are sometimes really hot or really acidic. I'm using some of those materials for a new project where I'm trying to use modern hot springs as analog systems for ancient ecosystems on Earth. So during 3 or 4 billion years ago, what did Earth look like and the microbes that were living there at the time.
So then how do the next steps of that process work after you go and collect the samples and either bring them to the university in Montana or bring them back here to Harvard? And tell me about how that process works.
Yeah, so it's just months and months of lab work, truly. So you can get the samples back, and then depending on exactly what you want out of them, it's basically a lot of lab processing to isolate the exact compound that you want to study or compounds that you want to study. So I oftentimes have looked at the lipids, and so I'll just talk about that. But basically, if you have like a hot spring sediment, you need to freeze dry it to get rid of all of the water. And then you do this extraction method where you get all of the organic matter out of the sediment that you have. And then you basically do a bunch of fancy chemistry steps to isolate the specific types of lipids that you want. And then what really ties together all of my graduate school work is the use of mass spectrometers. So those measure, the isotopic composition of your molecule. So again, how heavy are the atoms that are in that compound? So I basically do a bunch of like things that you learned in organic chemistry in the lab. And so I isolate like this specific class of compounds, and then I put them on this really fancy machine mass spec that then tells me like, oh, this is the isotopic composition of your molecule, or this is the type of molecule that's present, if that makes sense.
Yeah, absolutely. So to break that down even further, please correct me if I say something stupid here. Okay. You basically break the samples down, as you said, using different chemistry to get like one type of atom. So maybe like just trying to get only different types of oxygen. And you see how much oxygen of a certain weight there is in a ratio compared to oxygen of a different sort of weight. Is that about right?
No, I think specifically in the lipid realm, I'm looking at carbon isotopes. So what happens is if you have a sample of lipids, that gets sent to this mass spectrometer where those lipid molecules are ionized by a beam, and then it produces basically like this ion gas of carbon. And then that carbon gets sent to this mass spectrometer, which kind of like shapes the beam in certain ways, and then basically your light and your heavy carbon will get separated, and the machine will count how many instances of that light and heavy carbon you have and be like, yes, exactly what you said. This is the ratio that we have of the heavy and the light weights.
Cool.
Yeah.
Cool. Okay, so to recap, get samples, bring those back to lab, break them down, and then measure the ratios of different masses of elements. Is that right? It's
like measure the ratio of the different isotopes that are present, which are the elements that have different masses because they have different number of neutrons in their nuclei.
Perfect. Yeah. Thank you. Sorry.
No, it's a lot of complex stuff.
No, it is.
It's like, that was definitely something where I started grad school and then it was kind of like, yeah, you need to know how mass spec works. And so that was a little bit of a learning curve, right? Of understanding exactly what's going on in there. And then trying to communicate it in a like beginner friendly way is a little difficult, but yeah.
Sometimes concepts are just complicated. Like you need to, they're not always for beginners. Like you need to kind of dig into them a little bit.
Yeah, but I think it does make sense if you're like, oh, these things have different masses that affect how they kind of get separated in this machine, and then you can go and count them. So yeah, I think going from sample to actual like ionized carbon gas is the mental leap there potentially. Yeah.
So how has it gone for you so far? How have the samples been?
It's been super cool. Yeah, on the hot spring lipid side, I've gotten a data set out of there that is actually pretty interesting and promising because it's showing us that typical signatures of life that people have used in the past to be like, oh, if we go and measure the carbon isotopes in this ancient organic matter, this certain value looks like it is biological. So we can say that there was life back then. And then what this study is kind of trying to do is better characterize those windows that would be considered biological values. And it does seem like we're kind of expanding that through this research. So it's a preliminary data set that we have, and we're trying to get some more samples from more like diverse environments to try and really span that range of what looks biological versus abiological.
Cool. Okay, so you're trying to measure those windows of life. Is it just, I'm assuming you're trying to measure when that life was around in Yellowstone specifically, or wherever the samples are from? Is that the idea?
So yeah, it's interesting because I'm not one of these people, but there are some really cool people who go out and study, Earth's oldest rocks and that sort of thing. And they would be more on the like, hey, is there actually life in these rocks? What I'm more doing is I'm using Yellowstone as kind of this more constrained and modern system. What a lot of people would say is like a proxy system or an analog system, to say, hey, we have more constraints on these hot springs and we kind of think we know what's going on and we know that these microbes are present there. So let's see what their signatures look like because we can better characterize those and then apply that knowledge to things in the past to be able to say if there was life or not.
Cool.
If that makes sense.
And what's the end goal? So you're going to be writing papers, putting out posters. Tell me about your plans.
Great question. I was actually at a conference this past weekend with a poster for this project specifically. And the end goal for all of this is to write papers to put in peer-reviewed journals and all of that kind of stuff. So that people are like, yes, this is a new finding that we can take and read about and then develop on further. So yeah, with all of this stuff, it's to write papers, but again, second year PhD student. So it's like I have these posters that are kind of like, here's my preliminary results to tell the community before the maybe like final polished product comes out. And then I have this other project that is being started. It's starting to develop. I'm being trained on the machines and all that kind of stuff. So it's getting there.
Cool. Yeah, well, I want to hear about the new project as well, but I'll put a bow on the first one. So what do you think are going to be the lasting impacts of that first project? What are the end goals that you want people to take away from? And what do you want people to learn from what you've learned along the way?
Yeah, I mean, this is a project that I was super interested in. I kind of like came up with the idea with my advisor. And the idea for this is really to be more confident in when we actually go to the geologic record to say whether or not there was life or not. Because a lot of studies now are focusing on astrobiology and identifying life on other planets. And not that that's always necessarily from rock evidence. I think, Mars, we can maybe get some actual physical rock samples from there and be like, oh, cool, life. But a lot of people are doing gas forms of that, right? I am pretty much grounding myself on Earth, and I'm not thinking about necessarily those astrobiological implications yet. But it is important to think about when you go into the past, things are so jumbled that it's really good to have an important understanding of what we know is possible in the modern sense, and then go and try and apply that to the past, if that makes sense. And so it's interesting when you try and say, oh, this is evidence for the oldest life on Earth or something like that. That gets very contentious in the earth sciences. And when you're using carbon isotopes or maybe like physical characteristics of these like evidences of life, it's really hard to distinguish between what is biological and what is not, because Earth is crazy and like chemistry can produce so many signatures that are very similar to what biology does, because biology is also just chemistry and it's just harnessing these reactions that could happen without the presence of life. So being able to actually distinguish like, yeah, that's biological and that's not is really difficult and requires a lot of nuance. The purpose of this study is to add to some of those studies. It's definitely not like the end all be all of this, but it's okay, what do we know is a biological range? And then a really important part of this as a follow-up would be, what are some abiotic processes and what did those signatures look like? And then can we distinguish between those two? So that's more the end goal for this project in the long term, but it's just one component of that.
That's awesome. I saw also on the Harvard website, I don't know if this is still true. It said that you were a member of the Harvard Origins of Life Initiative.
Yes, yeah. So yeah, very cool organization. There's one at Harvard, and there's also like an international Origins of Life initiative, because I think people are just really interested in that at this point and thinking about life on other planets. And are we alone in the universe? Like really big philosophical questions that I think everybody thinks about every once in a while.
Cool. So yeah, tell me about this other project that you mentioned you're starting to get into the starting stages of now. What's that one about? What are the machines you're working with? And yeah, why is it important? What's the end goal?
Yeah, so this one is really trying to constrain the mechanism of pyrite oxidation, like that chemical reaction that I mentioned earlier. And this has implications for our understanding of like the evolution of Earth's surface throughout geologic time. And then also, again, for those modern like acid mine drainage impacted systems, because basically if you have a chunk of pyrite, it is weathered and broken down into these resultant products like sulfate, which can be preserved in the geologic record. So a lot of people will go and look at sulfate minerals and use their isotopic compositions as proxies for like, hey, what was the past concentration of oxygen in the Earth's atmosphere at that time, or CO2, or how large was the Earth's biosphere during that time, which are all really important for understanding like what did Earth look like 100 and something million years ago. And then on the other end, the mechanism of pyrite oxidation isn't super well understood. And so when you go to modern acid mine drainage systems today, there could be, different environmental conditions that change how that pyrite is reacting. And it actually changes the amount of acidity that is produced. So if you care about like, I don't know, what you have to do to the system to make it less acidic and to actually remediate a certain system, that can be really important, because we're also thinking about how microbes influence this process and whether or not they change the end products and the amount of acidity that's produced through this reaction. Yeah.
Cool.
I just remembered you asked me like, what machines I'm using. Again, mass spectrometers, but ones that have like different interfaces at the beginning.
Yeah.
So I have to be able to take like a solid sample to a gas. So it's pretty different from what I use downstairs on the actual like what the user is doing, but then it always goes to a mass spec at the end. That's kind of like the commonality.
Okay, sweet. And then what's the end goal of this one? Do you even know kind of what your goal is to be able to show with this project?
Yeah, so with this project, trying to use oxygen isotopes, so not carbon isotopes anymore, like moving into a different system, basically to be able to show where the oxygen in the resulting sulfate is coming from. So pyrite has the chemical formula like iron and then 2 sulfurs. And then through this oxidative weathering process, you're getting sulfate that has 4 oxygen atoms in it. And so some people think that like 3 of those oxygens come from water, 1 of them comes from like atmospheric oxygen, but that isn't actually super well constrained. So what we're hoping to do is to figure out exactly what that reaction process is and then where those oxygens are coming from. And so that will inform people who are doing that proxy development of like what those signatures actually mean, and then also to understand like how microbes are speeding up that process. And so I can't really say exactly what that's going to be yet, but definitely thinking about how microbes affect that reaction mechanism could be important for potential applications. But we're just trying to say this is the reaction mechanism and then think about the implications from there.
Cool. Okay. So then are the potential applications of that to, I guess, change the ratios of elements in our atmosphere? Is that where you're thinking to go with this?
It's to have better informed proxies to understand what Earth looked like in the past, would be the geological side of that. And then the other side would maybe be for people who are looking at really acidic systems that are impacted by acid mine drainage, to go and be like, okay, well, we know these kind of microbes are probably influencing this process in this way and all of that kind of stuff. But since I am in Earth and Planetary and I tend to think more about Earth's past, it would more to be like, okay, so now when we revisit this old data that's been produced, what do we think Earth's oxygen concentration was in the atmosphere a long time ago, or CO2 or something like that?
Yeah, and in that geologic perspective makes a lot more sense to me, and probably to you, I'm guessing, because that's where our background is, and like trying to, the best you can possibly constrain that data. And the best data you have in the past is really good for all of us here in the present. Tell me a little bit more about the other application. You said it was for acid mine drainage. I have no idea what that is.
Well, I'm not super familiar with those systems either, but I have just seen it in the literature where basically you can get these systems where people have gone and mined for different minerals, right? And they've basically exposed all of these rocks to the atmosphere or different water reservoirs or something. And so you're getting these reactions that are producing a lot of acidity. So if you have something like pyrite and it is weathered because you basically just dug a big hole in the ground and you exposed it to a bunch of oxygen, you're getting these reactions that produce hydrogen ions or like H plus ions, right? And that changes it and makes it more acidic. And then you can get runoff of these really acidic waters into like these really nice forests or something like that, where then you have die off of some trees or plants that can't handle acidity very well. So there's a big like remediation effort in a lot of those systems. I've gone to some of those during like field work. That was just for training purposes. Yeah. But yeah, it's definitely, I think, a big like environmental effort to remediate those acid mine drainage impacted systems.
Perfect.
Yeah.
And I think I just have two fun questions here. Cool. I saw, again, going back to the Harvard bio page. Yeah. I don't know if you even remember this picture that's up there, but the picture of you that's up there, it looks like you're kind of showing off a rock behind you.
Okay, yes. I don't have that many good geology pictures of myself.
I don't have anything like that, so this is a fun one.
Yeah. So that was actually, I'm showing off, like, I don't know if you could see it very well in the photo, but it's called a stromatolite. And so that's basically a like fossilized biofilm or microbial mat, as people would say. And that's oftentimes used as like evidence for the earliest life on Earth. You can go back in the geologic record and see stromatolites at like 3.5 billion years ago. And then people say, oh, cool, that means that there was like a biofilm or a microbial mat. And that means that life had been established by then. So it's like, it was my first love in geology and geobiology.
Cool. What exactly is a stromatolite?
Right. So a stromatolite is more of a like shape almost. Like you would say like a morphological feature, but it kind of looks a little wavy.
It is life though, right? It is some version of life.
It's not life. It's more like, you think about a fossil, right? Like when a dinosaur dies, it leaves behind its skeleton and we find it years later. Like some people could say it's like the imprint almost of a microbial mat. Cool. Because what the mat does is, you know, they're microbes and so they don't like leave behind a lot of fossil evidence. But what they do is they're really sticky, these biofilms. And so they trap a bunch of sediment onto them that kind of records this like trapping process. And they kind of grow and shift and move as they're trapping this sediment. It records that like morphological feature in the rock record. So it's like, it's not life, but it's basically this weird kind of fossil evidence of life.
Cool. Last fun question here. What's been your favorite class so far here at Harvard or undergrad?
Yeah, I think that one that wasn't super in my wheelhouse that I thought was really fun was Precambrian geology. Ooh. Yeah. So that's basically like the rock record up to 2.5 billion years ago. So it's kind of the oldest record of Earth history, right? And it's taught by a professor here who does study like the oldest rocks on Earth. And yeah, it was just a super cool class to think about like what Earth looked like that long ago. I don't know. It's kind of like, it's almost like storytelling, but it's so scientific that you're like, I don't know. It's just crazy to think about how different Earth looked back then. And I wasn't really super familiar with everything that was present in the rock record there. And so I think that kind of inspired my research project that I'm working on of being like, okay, analog system to what we see in that rock record. Because there's organic matter there are stromatolites and all of that kind of stuff where we know that life was present. But we don't know exactly what that life looked like or what it was doing. So it's just, it's really cool to think about.
Perfect.
Yeah.
Thank you so much for your time today, Amanda. Anything else you want to share with people? Anything else you want people to know and think about when they think about research?
When they think about research, I mean, it just does so much for us. So I obviously think about rocks, which people might say, oh, that doesn't have a lot of implications for people's daily lives, but it does inform climate models and all of these different things that are super important for human impacts. But I mean, research is so broad that, you know, I might be doing geochemistry, but everybody here is doing, I don't know, health impacting research of studying human disease and that sort of thing. So it really is this important creation of knowledge that I think is super important for not only human health, but also ecosystem health. And then, I don't know, just safety and people's lives. Yeah.
Yeah, totally agreed. Thanks again so much, Amanda.
Yeah, no,
Appreciate it.
absolutely.